We have our trends in manufacturing. And we're very pleased to be able to introduce our next speaker, Julie Audet. She's the Vice Dean of Graduate Studies, the Faculty of Applied Science and Engineering at the University of Toronto, with a very lengthy list of accommodations, which I won't get into for the sake of time, but welcome. Thank you very much for coming, and enjoy your talk.
Thank you very much for inviting me. So the title of my talk today is Model-Free and Model-Based Approaches for Cell Culture Optimization. So I'll be talking a lot about cell culture optimization. The specific challenges in cell manufacturing that the work that I will present today is addressing are indicated here. So there's a requirement in cell manufacturing for clinically and an industrially compliant process. So we need a consistent cell product, serum-free, xeno-free, chemically defined. So this results in a very large number of parameters to optimize in combination. So a lot of experimental work. Unfortunately, the gold standard cellular assay usually are not high throughput. So during the optimization process, one has to rely on surrogate assays. We need to minimize the uncertainty associated with these assays and the prescription from these assays in order to reduce the amount of experimental work with the labor-intensive assays. So the goal of my work related to this presentation is to significantly reduce the amount of wet lab work required for cell culture optimization using analytics, and ultimately tackle optimization problems that are currently unsolvable. So this is, in my view, the universal scheme for cell culture process optimization. So the first step is probably the most difficult. You have to come up with a list of factors of interest, the factors that you are interested in studying. And those factors, I will be talking mostly about soluble reagents, but then it could be also physical chemical parameters like pH or temperature or partial pressure of oxygen. And then so you have to decide on the range of doses, for instance, you are interested in covering for each of those factors if you're studying soluble factor. And then the next step is what I'm really interested in. So how do you decide on which factor combinations you will test? And based on the results that you get experimentally, how do you decide what to test next? So you have an iterative process. So at this step, there are several methods. So you can rely on best guess, intuition, and then, of course, there are more sophisticated methods, and this is what I will talk about today. So I classify the approaches mainly in two categories, model-free approaches and model-based approaches. And I'll give some examples of both in terms of the work that we have done. So first, I'll start with the model-free approach. And so we develop a cell culture optimization algorithm based on differential evolution. So this is based on evolutionary computing. So the principles were presented by Storn and Price in 1997, and it was used mostly to solve complex equations that could not be solved analytically. So we had to do a lot of work to use this algorithm. We had to make many modifications. I don't have time to talk about all the modifications to make it work for what we were interested in doing, which was to solve large cell culture optimization problems like 15 factors at 6 doses. So this is a very large parameter space. So basically, with differential evolution, quickly, you usually make combinations first randomly. You test them. and you select the ones that are performing the best. So if you are interested in maximizing cell expansion, so you will focus on the best combination, and then you create new combinations by randomly modifying those combinations. So this is called mutation, or recombining the best performing combinations. So this is called crossover. So the algorithm continues to cycle and create new generations until an acceptable solution is found. And this is analogous to walking on the response surface. So let's imagine that the true response surface of the process is unknown, usually, but let's say that it's represented by this surface in color. By differential evolution, you would be walking on the surface until you find the maximum of the function. So initially, we did some work with model cell line, and we had really good results to develop a serum-free culture media. So we were interested in testing the algorithm in a more relevant cell culture process, and we were extremely lucky that CCRM were interested in letting us test the algorithm with their T-cell process. So my PhD student, Michelle Kim, went there for a few months and tested the algorithm. And so we were able to compare the performance of new combinations with the current serum-containing media that CCRM was using at that time in their T-cell culture process. So once again, we were interested in developing a serum-free formulation. So we made a list of 14 factors at 16 doses. So this is more than 7 million possible combinations. And you can see the scores for combinations that were tested in each generation. So the process to its 6th generation. And so we were able to identify a few combinations, in this case, that scored close to the current serum-containing conditions, but ours were serum-free. And this is to give you an idea of the number of combinations that were tested in each generation. So this is a cumulative number. It's about 600 combinations. So we tested less than 10 to the minus 5% of all possible combinations to achieve that. And the code is published. So next, we did some repeat experiments with additional controls, and this reports the cell expansion obtained with the PC is the serum media used at CCRM, and then so it was ex vivo, I think, plus serum. The media without the serum, and then we tested the Xuri plus serum, Xuri media minus serum, and our 5 formulations. We did that for 3 donors. And this is the result that we have obtained. So we can see when the serum is removed from the commercial formulations, you see a dramatic drop in cell expansion. And this is compared with our serum-free formulation, which did supported expansion at a similar level to the serum-containing commercial formulation. Something that was interesting is that when we look at the variation from donor to donor, so usually when serum is removed from the commercial media, there's a huge increase in the coefficient of variation from donor to donor. However, in our formulation, the variability was lower. And this is the overall composition of the 5 formulations. So you can see it's rich in arginine, glutamine, interleukin-2. So next I will move to the model-based approaches for optimization. So just quickly, so these methods are based on design experiments. So the experiments are based on rigorous statistical principle. And then the second element of the approach is that the experimental data collected in those design experiments is fitted with a quadratic model. So the quadratic model describes the effects of the main factor effects, the effects of the interactions between factors, and also the nonlinear dose effects of the factors. So this is a simple model, quite rigid. Sometimes it works, sometimes it doesn't work. And I'm giving an example here where the model doesn't work so well because the true response surface is very complex, the topography is complex, and then the quadratic model is not a very good approximation. We used this method, we started using this method in collaboration with Nicolas Pinault's lab at Canadian Blood Services and University of Ottawa. So he was interested in developing a hemopoietic stem cell expansion culture media where with a small molecule cocktail. So this small molecule cocktail, the dose of each factors was going to be optimized, and this was going to be added to a serum-free media, which already contains stem cell factor, TPO, flat ligand, and LDL. So the factors are SR1, UM171, VPA, and an undisclosed factor. So I simplified the protocol quite a bit here, but the cells were cultivated for 14 days, and at the end, the cell phenotype, the number of CD34 positive, CD45 RA negative, PCR high was measured, and this was used as a surrogate stem cell assay. So first, we fitted the classic quadratic model that I mentioned before, and we had really bad results. So this method is very rigorous, and there are many statistical tests to validate the model, and some of them were failing. So we could see here in particular, we had, you see those outlier points. So this graph represents the experimental data, so the log ePCR expansion, and then the values predicted by the model. So normally you would want to have all the points. So this is the each factor combination in the factorial design experiment fall on the line. And then we had outliers in the region of interest, which is high ePCR cell expansion. And it turned out that those combinations were the one where UM171 was at the highest, and the other factors were at the mid-level. So it was very difficult at this point to use the model to predict what would be the best new combinations to test to maximize cell expansion, since it was clearly this model was overestimated the expansion obtained at high factor concentration. So we had to use a different approach. And so we used machine learning to try to obtain a better fitting model, a more predictive model. And you can see here the comparison between the classic quadratic that I started talking about that didn't work so well, and the machine learning model with the blue cross. So in terms of the training and validation, so the machine learning model did well. Interestingly, if you look at the prediction profiles for the quadratic and machine learning models, so those are those response curves for the 4 factors. So this is only one-dimensional. If you change the level of one factor, this will change slightly the shape of the dose response curve for the other factors because there are interactions. So you can see there's a difference in the way UM171 is modeled by the two, the quadratic and the machine learning model. And we use this computational tool to predict which new combinations of factors would be best to maximize ePCR cell expansion, so by interpolation. So because we had some uncertainty with the quadratic model and the machine learning model was a new method, we tested more combinations than necessary. And these are the results that were obtained. So 2 combinations stand out. what is interesting is that in machine learning, you have to use a test set, a completely independent data set to finalize validating your model. And because you want your model to be able to predict the outcome of future experiment. And this is what is represented here. So again, you can see the blue cross, the machine learning model, was good to predict the result of future independent experiment, while the quadratic model was really overestimating the results. So finally, we were after selecting a few combinations that will be tested with the gold standard assay later. So this is a step where you would want to minimize the number of combinations to be tested. So in retrospect, the two modeling approaches, the quadratic model and the machine learning model, predicted the same top two factor combinations. But of course, we couldn't really have a high confidence with the quadratic model because the predictions in terms of expansions were overestimated. So the machine learning model actually zoomed on the same two top combinations, and they are represented here. So the scale is not log transform, and those are the concentration. My final words, I think ideally an optimization method should actually combine model-free and model-based approaches because they are really complementary. The vision is to develop a versatile system actually that can decide which method is most appropriate based on the data that it's seen, and it can switch from one approach to the other. And this system could be completely autonomous. Things that would be interesting to do with this such a system is obtain on-demand formulations based on small molecules as opposed to recombinant cytokines that are more costly, or focus on clinically approved molecules. Also, if we make it very easy to develop serum-free, xeno-free media, it would be easier to do that early in the developmental process instead of doing that later and repeating many experiments with a new media that's chemically defined. So finally, I'd like to acknowledge those that have participated in this work. So Michelle Kim, a former PhD student in my lab, we received funding from NSERC, and Cecilia RM was funded by FedEv Ontario for this project. And then, of course, Nicolas Pinot's lab works on hemopoietic stem cells, and his group. And we received funding from Canadian Blood Services for this work. Thank you.